Veteran Jakarta mengukuhkan diri sebagai kampus bela negara yang merupakan identitas tak terpisahkan sejak kampus ini berdiri tahun 1958 hingga mencapai status universitas negeri pada tahun 2014. Sejak itu, transformasi terus bergulir.
UPN Veteran Jakarta sebagai PTN adalah merupakan akhir dari proses panjang tentang kejelasan status. Dengan menjadi PTN, maka ada kepastian tentang status tersebut, dan selanjutnya yang terpenting saat ini adalah bagaimana UPN Veteran Jakarta mampu untuk sejajar dengan PTN-PTN lainnya. Dan tentu saja yang terutama adalah bagaimana UPN Veteran Jakarta dapat memberikan kontribusi dan pencapaian program pemerintah khususnya di dalam pendidikan tinggi. Dalam hal ini adalah program dari Kemenristek-Dikti.
Bela negara menjadi rujukan dalam penyusunan kurikulum demi mewujudkan visi UPN Veteran Jakarta, yaitu menuju perguruan tinggi berbadan hukum, inovatif, berdaya saing yang beridentitas bela negara tahun 2025.
Dalam rangka mentransformasi UPN Veteran Jakarta menjadi PTN yang unggul dan berdaya saing di tingkat nasional, kerjasama menjadi faktor yang sangat penting bagi kita. Kita selama 4 tahun terakhir itu selalu masuk dalam posisi 10 besar. Jadi kualitas mahasiswa yang masuk di UPN ini mahasiswa yang cerdas dan sangat cerdas. Jadi secara keseluruhan upaya peningkatan kualitas pembelajaran kita lakukan secara sistematik, mulai dari kultur akademik, kemudian desain kurikulumnya, sampai peningkatan kualitas SDM kita hingga sarana prasarana pembelajaran.
Didukung dosen dan tenaga kependidikan yang handal, UPN Veteran Jakarta siap menjadikan setiap lulusannya mumpuni dalam 7 fakultas yang tersedia. Fakultas Ilmu Sosial dan Ilmu Politik,
dia masih perkenalan kamu.
Fakultas Hukum, Fakultas Ilmu Komputer, Fakultas Kedokteran, Fakultas Teknik, Fakultas Ilmu Kesehatan, serta Fakultas Ekonomi dan Bisnis. Sementara untuk menunjang kegiatan akademik, UPN Veteran Jakarta menyediakan pusat bahasa atau Labimkar, LBH atau hukum, sarana ibadah, poliklinik, laboratorium teknik, laboratorium kedokteran, laboratorium stem cell, laboratorium penunjang lainnya, perpustakaan, sarana olahraga, dan keamanan.
Di dalam mendukung daya saing para mahasiswa di dalam menempuh proses belajar mengajar di UPN Veteran Jakarta, UPN Veteran Jakarta telah memiliki beberapa sarana prasarana yang dapat digunakan sebagai salah satu peningkatan daya saing. Antara lain adalah perpustakaan yang sudah online, ya, berbasis dengan digital 4.0, di mana para mahasiswa saat ini bisa memperoleh referensi melalui online, jurnal-jurnal melalui online. Demikian juga sarana prasarana pendukung, kita memiliki Mesh Classroom. Di sini digunakan Mesh Classroom ini adalah suatu kelas yang dapat dilaksanakan kuliah secara bersamaan dengan teknologi yang canggih, di mana para mahasiswa dapat mengikuti dan para mahasiswa secara langsung dapat berinteraksi dari tenaga pengajarnya yang ada di satu tempat. Ya, sarana ini ada di kampus Pondok Labu maupun di kampus Limo.
Berbagai kerjasama dengan lintas pihak terus dilakukan untuk meningkatkan kualitas pendidikan dan meningkatkan lulusan pada kerja nyata. Sementara untuk meningkatkan kapasitas non-akademik, mahasiswa difasilitasi 25 unit kegiatan mahasiswa yang dapat dipilih sesuai minat.
Program-program yang kita terapkan di UPN Veteran Jakarta kepada mahasiswa pada dasarnya adalah peningkatan kualitas kegiatan kemahasiswaan yang nantinya akan meningkatkan rasa cinta kepada tanah air, rela berkorban demi nusa dan bangsa, dan juga berjiwa bela negara. Peningkatan rasa cinta tanah air itu diwujudkan dalam kegiatan kemahasiswaan yang sungguh-sungguh, kemudian mencapai prestasi yang tinggi baik di tingkat nasional dan juga internasional.
Berbagai kelengkapan sarana hingga prasarana tersebut telah menjadikan UPN Veteran Jakarta sebagai kampus rujukan generasi cinta negara. Tingkat kepercayaan di kalangan calon mahasiswa terus tumbuh. Animo pelajar untuk berkuliah di UPN Veteran Jakarta terus meningkat. Hal ini dapat dilihat dari masuknya 2 program studi UPN Veteran Jakarta dalam 10 prodi sosial dan humaniora dengan peminat terbanyak di Indonesia. Program studi ilmu komunikasi menempati urutan kedua dengan peminat sebanyak 2.981 calon-calon mahasiswa. Sementara program studi manajemen berada di urutan ketujuh dengan peminat 2.532 calon mahasiswa. Selain itu, nilai rata-rata calon mahasiswa yang masuk ke UPN Veteran Jakarta untuk program studi sains dan teknologi berada di urutan ke-10 dari perguruan tinggi di seluruh Indonesia. Berbagai prestasi tersebut telah menjadikan kampus UPN Veteran Jakarta sebagai salah satu kampus unggulan di Indonesia. Dalam 3 tahun terakhir, peringkat kampus secara nasional terus meningkat. Kemenriset Dikti mencatat di tahun 2018, UPN Veteran Jakarta berada di peringkat 143, meningkat 290 angka dari tahun 2017. Pada tahun 2019, UPN Veteran Jakarta masuk 100 kampus unggulan nasional atau berada di peringkat ke-77. Transformasi UPN Veteran Jakarta yang telah menjadi universitas negeri adalah tonggak awal menuju era baru, mewujudkan universitas terkemuka di Indonesia.
Selamat berkarya, maju bersama, meraih prestasi.
Salam bela negara.
Okay, we start now. Okay, good afternoon, ladies and gentlemen. It is my pleasure to welcome you to UPN Veteran Jakarta International Webinar with theme SARS-CoV-2 from Virology, Molecular Biology, and Clinical Pathology Perspective. The Honorable Rector Universitas Pembangunan Nasional Veteran Jakarta, Dr. Erna Hernawati, AK CPMA CA, the Honorable Dean Medical Faculty of Universitas Pembangunan Nasional Veteran Jakarta, Dr. Priyo Sidi Pratomo, Specialist Radiology Consultant, the Honorable Speaker, Khalid Al-Kharsah, PhD from College of Medicine, Imam Abdul Rahman bin Faisal University, Arabia, Seok Muing Wang, PhD, University Technology Marat, Malaysia, and Dr. Sore Lardo, Specialist Penyakit Dalam, Finasim, Faculty of Medicine, Universitas Pembangunan Nasional Veteran Jakarta, and also Gatot Subroto Army Central Hospital, RSPAD, and also the Honorable Moderator, Dr. Yudi Nugraha, MBiomed, and all colleagues and all webinar participants from around the world. I welcome you in this international webinar. First, before we start this event, we have to listen first from national anthem from Indonesia. The participant, please raise for IT. Indonesia Raya, please, song the Indonesia Raya. Okay, thank you. So, and we came the next step. First of all, let us pray and praise to God, because if His bless and mercy, we can come together without any obstacle here with healthy condition in the seminar. Please, I introduce myself. My name is Andri Pramishanti Pramono, PhD. I will be your host today. For those of you who don't know me, I'm the head of medical research and also the chief editor of medical journal in medical faculty, Universitas Pembangunan Nasional Veteran Jakarta. It's such an honor to welcome all of you here today. Ladies and gentlemen, on this special event, we have several agendas as follows. First, we have opening speech from our rector, Universitas Pembangunan Nasional Veteran Jakarta, Dr. Erna Hernawati, AK CPMA CA. The second one is the welcoming speech from Dr. Priyo Pratomo, Specialist Radiology Consultant, Magister Law, as Dean of Medical Faculty, Universitas Pembangunan Nasional Veteran Jakarta. The third, we come to the main lecture is from Khaled Arhasa. The first one from Department of Microbiology, College of Medicine, Imam Abdul Rahman bin Faisal, University, Saudi Arabia. And then followed by Selmi Wang, PhD. She was the Associate Professor, Faculty of Medicine, University Teknologi Mara, Malaysia, with title Understanding Molecular Pathway of SARS-CoV-2. And the last lecture will be presented by Dr. Soroy Lardo, Specialist Penyakit Dalam Finasim from Faculty of Medicine, Universitas Pembangunan Nasional Veteran Jakarta, Gatot Subroto Army Central Hospital RSPAD, with title Clinical Perspective and Community of SARS-CoV-2. The last, we will have a discussion and time for the participant to give question to the speaker, and the closing one. Now we invite our respectful Rector Universitas Pembangunan Nasional Veteran Jakarta, Dr. Erna Hernawati, AK CPM ACA, to give an opening speech. To Dr. Erna Hernawati, time is yours, please.
Okay, thank you, Bu Andri. The Honorable Dean of Medical Faculty UPN Veteran Jakarta, Dr. Priyo CD Pratomo, Specialist Radiology, Magister of Law. The speaker, Mr. Khalid R. Alkasa, PhD, Virology of SARS-CoV-2, Associate Professor, Department of Microbiology, College of Medicine, Imam Abdul Rahman bin Faisal, University, Arab Saudi. Thank you. Ms. Wang Seok Mui, PhD, Associate Professor, Faculty Medicine, University Teknologi Mara, Malaysia, and the Honorable Dr. Soroy Lardo, Spesialis Penyakit Dalam Finasim, as a lecturer Universitas Pembangunan Nasional Veteran Jakarta, and all participants. Assalamualaikum warahmatullahi wabarakatuh. Greeting to all. Praise be to Allah Subhanahu wa Ta'ala, Almighty God, for His grace and grace today. We can still gather in the study program, even though in the COVID-19 pandemic. Hopefully, COVID-19 will pass quickly and we can live normal again. Amin, amin ya rabbal alamin. This event become very valuable with the presence of the all excellent speakers from Saudi Arabia, Malaysia, and this seminar is special because it discusses COVID-19 from various perspectives. Moreover, having speakers presenting from 3 countries is not easy, I think. But in fact, during COVID-19 pandemic, this has become possible. I think this is one of so many lessons and improvement we can gain during COVID-19 pandemic. Actually, anything will always have 2 sides, I think, negative and positive. It is only a matter of how we treat any event that occurs to us. I hope we are always able to take the positive side, even in the hardest time. I would like to also give highest appreciation to the Dean and his staff who are still enthusiastic to do something remarkable during pandemic. This is in line with our commitment that even pandemic must not be a barrier from being productive. All academic activities must continue. Lecture activities, examination, and academic services should and must run as usual with maximum effort with practicing appropriate health protocol. Ladies and gentlemen, related to COVID-19, as we know that COVID-19 pandemic is still going. In all over the world, some have shown a declining trend. while some still showing an increasing trend. In Indonesia, currently in its capital city, Jakarta, increasing trend still shows in the last few weeks. Government spokesperson for coronavirus, that Mr. Ahmad Yuryanto said that experts from all over the world were still trying to find a vaccine for the prevention of COVID-19. All parties continues to trying to find the COVID-19 vaccine. In fact, according to him, Indonesian researchers are also trying to make the discovery. Related to this condition, it is the duty of academics to continue to work on researching vaccine formula, possibility which can be helped to prevent COVID-19 transmission, and also guiding the community by socializing on what must be done in order to minimize the possibility of getting affected by COVID-19. So the most important things right now is how we try to protect ourselves. The whole community must make effort to protect themselves from COVID-19, because protecting people from COVID-19 is not only the duty of the government. Finally, congratulations on joining this seminar. Hopefully you will produce something useful and can also help to contribute to on COVID-19 countermeasure. Hopefully this collaboration between University Mara Malaysia and Imam Abdurrahman bin Faisal University from Saudi Arabia and UPN Veteran Jakarta will continue, especially in this pandemic. There are many things we can do through webinars like this, or maybe for the future we can join research. We have to keep us the enthusiasm and positivism to proactively to fight COVID-19 by always practicing health protocol as per recommended. May you stay safe, protected, and healthy during this tough time. See you. Salam bela negara. Wabillahi taufik wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Salam bela negara. Thank you very much for the speech, Dr. Erna Hernawati, AKCPM ACA, for encouraging speech. For the next one is welcoming speech from our Dean of Medical Faculty, Universitas Pembangunan Nasional Veteran Jakarta, Dr. Priyo Sidik Pratomo, Specialist Radiology Consultant, Magister Law. Please, time is yours.
Thank you, Bu Andri, as a moderator in this seminar. Do I have to continue with open the meeting, or I have to give it again to somebody, maybe director, to open?
No.
Oh, I directly, Bu Andri.
Yes,
Okay.
Dr. Priyo.
Good afternoon, and of course, good morning, because I have one speaker from Saudi Arabia still in the morning. Respected President of the University Pembangunan Nasional, Dr. Erna Hernawati, Honorable Speaker, Associate Professor Khalid Al-Karsah from University Imam Abdurrahman bin Faisal in Saudi Arabia, and then Associate Professor Mui Wang from University Technology Mara in Malaysia, and of course my colleagues, Dr. Soro Elardo from University Pembangunan Nasional Veteran Jakarta. First of all, I would like to thank you to all of you, especially Madam Erna Hernawati as a president of this university that have already given a talk or introduction about our university. So our university, maybe we can say, as a campus of defend the country. That is the motto for our campus. So during this pandemic, we regularly make the seminar, and this is already the second time in regard to the international seminar. The first one, we collaborated with the speaker or the colleagues from Japan. And now we make a collaboration again, but this time with the Saudi Arabia and Malaysia. I hope that all of you can share your knowledge, your experience, because in this time of seminar, we try to explore regarding the SARS-CoV-2 from the virology until to the perspective of community and also the clinical. I'm hoping that this knowledge will enhance to all of the people who will hear about this seminar, and of course to all of us here in Indonesia is a part of our combating of this COVID-19, because we are now still in the pandemic, and the disease itself is still continue, not yet regress. I hope that you can enjoy this meeting, and as a word of Bismillahirrahmanirrahim, I do declare this seminar is open. Thank you.
Thank you very much, Dr. Priyo, for the speech. So now we come into the main point in this seminar. It is my pleasure to introduce our moderator today, who will guide our talk today, Dr. Yudi Nugraha, M. Biomed. I will read a short brief of his CV. He has Bachelor of Science Biology from UPN Jakarta on 2011 and got Master in Biomedical Science on 2014 from Faculty of Medicine Universitas Indonesia, and have Membuka Gusho awardee from Japan government, and got his PhD in structural biology on 2018. And now he was head of stem cell and regenerative medicine laboratory in Universitas Pembangunan Nasional Veteran Jakarta. Okay, Dr. Yudi, time is yours. Please.
Okay, salam bela negara. Thank you, Dr. Andri, for a brief introduction of me. Firstly, again, we'd like to thank you and give our highest gratitude to our beloved Director, UPN Veteran Jakarta, Dr. Erna Hernawati, and our famous Dean, Faculty of Medicine, Dr. Priyo Sidi Pratomo, Specialist Radiology Clinic, Consultant, Master of Law, for their huge support for this international webinar. Actually, this program is a follow-up of international collaboration between UPN Veteran Jakarta and UITM that initiated by Vice Rector of Student and Corporation, Dr. Ria Maria Teresa, Spesialis Kesehatan Jiwa, and also initiative next international collaboration with Imam Abdul Rahman bin Faisal University. Actually, I was very impressed at first by the vibrancy of the audience. We accept more than 340 registrants in just 5 hours after register was open. The participants not only from Indonesia, but also from abroad, including Saudi Arabia, Germany, and Malaysia. So we make live shows through our YouTube channel, and we will give an e-certificate to audience by writing their full name in the YouTube comment and also in the chat. We'd like to thank you and express our sincere gratitude to all the speakers, Dr. Khalid, Sukron Jazilan, Dr. Wang, and Dr. Soroy, for giving their precious time to educate us about the pandemic that we have been going through for almost 4 months. We really have chosen the speaker based on their expertise and knowledge, of course. Hopefully, this webinar will have significant for us to deal with this pandemic. We already know that SARS-CoV-2 is a pandemic that has killed more than 370,000 people in the world and have been 6 million cases confirmed. In Indonesia itself, COVID-19 was killed over 1,600 people with 28,000 cases, and this number is still increasing. So with this interesting fact and purpose in dealing with the next new normal, we strive to initiate the discussion about SARS-CoV-2 with the various perspectives from virologists, molecular biologists, and clinical pathologists. The first perspective will be discussed from the fundamental virology itself. It will be given by our first speaker, Dr. Khalid Al-Karshah. Dr. Khalid Al-Karshah was associate professor and researcher from Saudi Arabia. He belonged to College of Medicine, Imam Abdul Rahman bin Faisal University, after graduating from Hanover Medical School in Germany. Then, next, we will continue to understand SARS-CoV-2 at the molecular level mechanism. And this lecture will be given by Dr. Wang Seok Mui. She is an associate professor and lecturer from the Faculty of Medicine, University Teknologi Mara, UITM, Malaysia. And the last, in this international webinar, we will also explain as to the clinical perspective and community of SARS-CoV-2 as close important statement lecture, and will given by Dr. Soroy Lardo, spesialis penyakit dalam. Dr. Soroy is lecturer and researcher at the Faculty of Medicine UPN Veteran Jakarta. He also worked at Gatot Subroto Army Central Hospital, RSPAD Presidential Hospital. So, to start this webinar, Dr. Khalid will begin his lecture on virology from SARS-CoV-2. So, Dr. Khalid, since now time is yours.
Thank you very much, Dr. Yudi. Just let me start with sharing the slides, and then thank you very much actually for inviting me for this webinar. I'd like to start by thanking Dr. Erna, the rector, and Dr. Rio, the Dean of the Faculty of Medicine. Thank you very much, and Dr. Andrew, of course, for inviting me for this webinar. It's my pleasure to be part of it. And I hope my talk will be useful and helpful in your final goals of this webinar. Actually, since I don't know what will be the spectrum of specialties of the speakers, I made the talk more general in the form of like general talk about virology. So I'll be talking about the family of the coronaviridae, where the virus is coming from, and I'll try to explain to you how does a new virus emerge, how new viruses appear in the human population, and I try to answer this question, which is the $1 million question: is this virus a naturally occurring virus, or it's a man-made virus? I'll talk briefly now about the genome structure and virion structure and replication, because Dr. Wang is going to talk about them. And then I'll just mention about the transmission, protection, and laboratory diagnosis of the virus. So the story starts, as you all know, actually in December 2019, where several local health facilities from China reported the presence of clusters of patients with pneumonia, and this pneumonia had no known viral or bacterial cause, but they were all epidemiologically related to the seafood and wet animal wholesale market in Wuhan, and this is the picture. So in December 31st, the Chinese CDC conducted epidemiologic and etiologic investigation, and in early January, they announced that these clusters of pneumonia are caused by a new virus, and they called it at that time 2019 new coronavirus. And I think by January 7th, the WHO announced the presence of a new virus causing pneumonia. And from then, the virus infected or spread to about 216 countries worldwide and territories, of course, and infected more than 6 million people, and unfortunately has killed so far more than 379,000. And unfortunately, the counter is still going on, and the numbers are growing. So what is this family of coronaviruses, the family Coronaviridae? It's a larger family of viruses that contains human as well as animal viruses. I think you all know now why it is called coronaviruses, because the virus has its solar corona appearance under the electron microscope, and this is how it looks like when it's stained with special stains, because of the it gets this solar corona appearance because of the S protein on the surface of the virus, as you will hear later in the talk. So the coronavirus family, Coronaviridae, contains 2 subfamilies and 5 genera. These 3 genera, they don't contain any non-human virus so far, and we hope they stay like that. We don't want to hear about any more human viruses from this family. But these 2 genera, the alpha coronavirus and the beta coronavirus, contain many human viruses. The first to be discovered and identified were the human coronavirus OC43 and the human coronavirus 229E. They were discovered in the middle of 1960s, and they cause only moderate or mild common cold or moderate respiratory tract infections. So these viruses, they were actually not very well studied since that time, and they were not given too much of attention until the appearance of the emergence of the SARS coronavirus in 2002 in China. Then that caused the pandemic also, but it was a limited pandemic. This family of viruses get a lot of attention and start to be investigated. And then researchers found actually that there are 2 other viruses in 2004, for example, the NL63, and in 2005, the human coronavirus Hong Kong University one. But also these viruses were found to cause a mild pneumonia and mild upper respiratory tract infection. Then actually in 2012, we were surprised to get a new virus from this family emerged in the Middle East, and it was called MERS coronavirus, and that was a severe virus. And then now we have, lastly, and hopefully this is the last one, but we don't expect it to be the last one, the SARS coronavirus 2, which emerged by the end of 2019. So these viruses in green are actually the viruses from human viruses from Coronaviridae family that they cause a mild disease, but the one in red are the nasty viruses. So what are the difference between or similarities between these 3 dangerous human coronaviruses? They all actually came from bat. Bat was the primary source of these viruses. They had different intermediate hosts. The SARS coronavirus, which emerged in 2002, the intermediate host was the palm sedate. And from there, it jumped to human. And this virus had a moderate human-to-human transmission. It infected about 8,000 cases worldwide, and actually it disappeared in 2004. We did not hear about this virus anymore, and it had about a 10% mortality rate. While the MERS coronavirus, which appeared in 2012, it had a dromedary camel as an intermediate host, and also this reported luckily less human-to-human transmission. This virus was highly lethal. The mortality rate was about 35%. It reached actually 40% at the beginning of the disease. But these viruses, we call them zoonotic viruses because they came from animal to human and did not establish a very well documented human-to-human transmission. In case of MERS, we had only clusters of human transmission among healthcare workers, but not more than that. Now, the 2019 emerging coronavirus, the SARS coronavirus 2, also came from bat, as I will show you later. The intermediate host is not yet well defined. We think it is the pangolin, but it is not yet well established. This has to be still investigated. This virus differs from other coronaviruses by that it has a very well established human-to-human transmission, and this is actually evident by the millions of people who are infected. but luckily it differs in that it has a lower mortality rate. So the average mortality rate is about 3 to 5%, and it differed according to between countries. For example, in Italy, it reached about 10%. In other countries such as Germany and Saudi Arabia, also here we have less than 1% mortality rate, alhamdulillah. So before I continue actually with the talk, I'd like to explain this because sometimes we mix the terminologies. The two previous coronaviruses were given the name of the disease. So in 2002, the severe acute respiratory syndrome given the name to the virus, so the virus was called SARS coronavirus. In the Middle East, respiratory syndrome was also given the name for the virus, and the virus was called MERS-CoV. In this new virus, they wanted to confuse us a bit, so the WHO called the disease coronavirus 2019, COVID-19, while the Committee on Virus Nomenclature called the virus SARS-CoV-2, as it is very similar to the SARS-CoV-1. So when we talk about the disease, we mention COVID-19. When we talk about the virus, we mention SARS-CoV-2. And since I will be talking about the virus, you will hear me always talking about SARS-CoV-2. So now, why and how do new viruses emerge in the human population? Actually, we have heard about many pandemics previously in the history, and the most common and famous one is the 1918 influenza pandemic, which killed actually more than 20 to 50 million people, unfortunately, at that time. And now we have the bad pandemic, the SARS coronavirus 2. There are different ways how a new virus might emerge in human population. The first way is that our non-human viruses, they mutate and produce a new virus. And this is very well known in influenza viruses, where they always develop a new strain. Influenza viruses, for example, mutate in two ways. One way we call it antigenic drift, and this is the gradual mutation that accumulate in the virus upon replication, and this is the mutation that is responsible for antigenic differences in the virus, and this is why we have a seasonal influenza every year, and this is why we have to take the vaccine every year against the virus. The other type of mutation, and this mutation actually, the antigenic drift, is because of the mistakes that the polymerase of the virus make while replicating the viral genome. Like the other method is called antigenic shift, and this happens when two viruses from two different hosts, they meet in one host and make a new completely virus by the reassortment of the viral genomes, and this virus usually is very lethal to human population. So this is one method. The other method is that an animal virus jump into the human population and become a human virus. And this is what we call a spillover. And this spillover might happen for many reasons. First of all, because there might be climate and environmental changes that change the habitat of the animal and bring them to the human life. And also it might happen because a human is invading the wild habitat of the animal. So we are going to forest, we are cutting forest, we are coming more closer to wild animal. We are using the wild animal. Some people are eating these wild animal, and this gives a chance for these viruses to jump into a human population. And also the increasing raising of animal near to human habitat. So we are raising now animals near to the cities, and this also gives a chance to the animal viruses to jump into the human population. So is that what happens? So for a spillover to happen, you need an animal primary source, which can be bat, bird, or rodent, or many other animals actually. And in this animal, you need a virus that is mutating and producing a new viral mutant. And then when this virus, the new virus that mutated in this animal, gets a chance to jump into the human population, it will become a new virus for a human, and it will cause severe disease. But luckily, this does not happen directly. It happens rarely directly like that from an animal directly into the human population because of the great differences in the host factors between the two hosts. It usually needs an intermediate host, which is usually also a mammalian host, where this mutated virus will develop further into this mammalian host, and then it will become ready to jump into the human population when it is possible. So again, the pangolin for SARS-CoV-2 is not yet confirmed to be the intermediate host. We are still looking, people are still looking for the intermediate host for this virus. So what I want to say also here that the primary virus here, the human virus, will not be similar to the primary virus in the bat. It will not be identical, sorry. It will be similar, but it will not be completely the same virus because it is actually mutating into the intermediate host. Now, the actually important question that has been discussed very much in the media and social media, and people are talking about it, is that SARS coronavirus, SARS-CoV-2, a human-made virus or naturally occurring virus? It's actually really difficult to answer this question, but we will talk about the evidence that we have. First of all, we know that viruses mutate, and we know that the recombination event happens between viruses. And this is also not uncommon for coronaviruses. So also recombination events happens among coronaviruses. Then we have also several studies which showed that this new coronavirus is most probably a result of a recombination event. For example, this is the first paper that was published about the virus, announcing that the virus is causing these cases of pneumonia. They showed here that the virus, in the genetic mutation, in the genetic phylogenetic analysis of the virus, that the virus actually localizes itself with the bat viruses in the same arm with the SARS coronavirus 1. And if this virus is actually man-made, why not SARS-1 is also a man-made? Actually, both of them are viruses that emerged by recombination, natural recombination in the nature. This is also another paper which was published very early about the virus, describing the virus genome. This paper shows clearly that that the virus, when they put the virus sequences in the phylogenetic analysis, that this virus sequence has changed the topology of the phylogenetic tree. And here they suggest that a recombination has occurred early in the past between these viruses. This is another paper that shows that there are more than 380 amino acid substitutions in the human genome, and this is not one single substitution, it's multiple substitutions in the viral genome, and this is actually really difficult. It's close to impossible to be done by a laboratory. This is also another study which showed that the virus is not a mosaic, and it consists of, in half of its genome, from a distinct lineage from beta coronaviruses. So it is not a virus that is very similar, it's a separate lineage of viruses. And also this paper here recently appeared and showed that this virus has emerged in a recombination event and strongly purifying selection. So after the virus mutates and appears by recombination, then it has to go through selection. Either it is a bad virus and it will die out, it will not appear, or it is good and fit virus, and then it will have a chance to survive in the host and then also jump into another host. So this is the last evidence also. It's a paper at the end shows that also that our analysis clearly show that the SARS-CoV-2 is not a laboratory construct or a purposefully manipulated virus. So these, I think, are enough evidence to show that the new virus, the SARS-CoV-2 virus, is most probably a recombination event, naturally occurring virus, and not a lab manipulated virus. So I'll briefly talk about the structure of the virus genome because Dr. Wang also is talking about it. So as I said, in the phylogenetic analysis of the virus, it localized the virus with the bat viruses. So it has about 89.1 similarity to these bat viruses, and it has about 97 similarity to the SARS-CoV-1. This is why it is called SARS-CoV-2, because it's actually very related to the SARS-CoV-1. While it is far away from the MERS-CoV, it has only about 50% similarity with this virus. So this is how the viral genome look like. As I said, I'll just go briefly through it. It is a single-stranded RNA. It has 14 open reading frames. These open reading frames produce about 27 proteins. The larger gene is the replicase, which has two ORF1A, ORF1B, and this produces 16 non-structural proteins that are important for the virus replication and production of virus particles. And then there are many other important proteins like the spike, EM, and nucleocapsid that we will hear about them later. So this is how the virus looks like. This is a very complicated picture or diagram of the virus with all of its proteins, but I'll take you to the more illustrative diagram. The virus is enveloped, so it has an envelope. It has a cover of a lipid bilayer. The virus has a helical capsid, the capsid where the RNA of the virus is located. It is long and helical. This is the capsid of the virus, and it contains the single-stranded RNA of the virus. And then on the envelope, you can see the viral proteins, the E protein, the M protein, which is an interferon inhibitor, and also the very important protein, which is the spike protein, which is important for the entry and binding to the cell and entry to the cell. So this is the spike protein again. It's a club-like or betel-like shape. This is how it looks like. It is always present in 3 copies together. Trimerization of the protein is important for its function. It is important for the binding and fusion of the virus with the cell receptor. Before binding the virus, this protein has to be cleaved actually by a cellular protease called furin at this side, and also it has to be cleaved here by another cellular protease called TMPRSS2. I think Dr. Wang will talk about that. These cleavage events are very important for the fusion of the virus with the cellular receptor. So by now, I think we all know also that the receptor for this virus is the angiotensin converting enzyme 2, the ACE2. And this is, the ACE2 is a carboxypeptidase, and it has a function for human in the cardiac function, and it's involved in regulation of cardiac function and blood pressure. Although it is enzyme, it's an enzymatic activity actually is not required for the entry of the virus. So if you inhibit the enzymatic activity of the ACE2, the SARS-CoV-2 virus still will be able to bind to the receptor and will enter the cell. So this receptor is present in the epithelial cells in the lung, small intestine, heart, kidneys, and upper respiratory tract, and many other tissues. And this is, of course, is simulate what is the, or tell us about the severity, about the disease or the clinical symptoms that appears on the patient in the upper respiratory tract infection, lower respiratory tract symptoms. We know that some patient develops diarrhea and the virus was detected in stool. And we know also that the virus causes heart diseases and kidney failure. So the distribution of the receptor over the human tissues tells where the virus is going to replicate. How the virus replicates it after binding and into the receptor and the entry of the virus, then the virus releases its genome, and the genome will be first translated into the first enzyme, which is the replicase enzyme, which will encode about 16 non-structural proteins. These proteins are mostly enzymes that are important for the other expression of the other viral proteins. Then the other viral protein will be translated will be then produced, and the viral RNA also will be then replicated. Then the viral RNA with the viral protein will assemble together, produce the new virus, and the new virus will be released from cells. And of course, here in the life cycle, we have a lot of druggable targets of the virus, like the binding, the enzymes here in the replication, the release, and all of these are possible target. I think Dr. Wang will talk about them, but unfortunately, we need to find the target for that can, the drug that can target these events in the virus replication, and then we have to test it and see that if it is effective or and safe on the human population. And this, of course, will take some time. So what about virus transmission? I think you all know now that the virus transmits through the droplets, the aerosols that are spread by the infected individual, infected person. Now we know that the virus migrate with the smaller droplets actually, which can migrate for up to 2 meters. So now the safe distance from a patient or from a person who is infected is 2 meters. At the beginning of the pandemic, it was 1 meter. Now we know that it is, 2 meters. So you can get infected if either directly from the person, from the infected person, or you can get infected indirectly if you touch the utensils or the fomite that the patient is using. And unfortunately, it's not the patient who is symptomatic is transmitting the disease. Now we know that patients who are also asymptomatic without any symptoms, also they can transmit the disease. Also, there are patients who are also pre-symptomatic. They are also possible to a transmitter of the disease. So this is, I think, the major problem in the transmission. The asymptomatic people, asymptomatic individuals, and also the pre-symptomatic individuals are also very important transmitter, very important to localize and find and to control the infection of the SARS-CoV or to control the spread of the SARS-CoV. And this is why now mask is very important. So mass masking is now very important because we know that there are a lot of asymptomatic transmission and pre-symptomatic transmission. So how you protect yourself from this virus? Actually, it is now known. You have to avoid any contact with the infected individual. You have to practice good health hygienes, like washing hands always, avoid touching the face, nose, eyes, and mouth, and also wear a mask whenever you go outside. And also, if you feel sick, you always directly go seek medical help. Don't delay it because the symptom will develop. And we all have to develop this cough etiquette, cough and sneezing etiquette. So if we will cough, always we use a tissue and dispose the tissue in a safe way. Or if we don't have a tissue, then we use the elbow of the arm. And of course, now there's no traveling, but of course, try to avoid traveling even locally nowadays to minimize the transmission of the virus. And since we don't know exactly the intermediate host, it's better to avoid contact with the wild animals. Now I'll talk briefly about the laboratory diagnosis of the virus, because I think Dr. Soroyo will talk about the clinical manifestation of the virus. The laboratory diagnosis, the laboratory detection of the virus is done by the gold standard method, which is the real-time reverse transcriptase polymerase chain reaction. I'm not going to talk here about the assay per se, because I think it's a lot of technical details, but I'll just say what we look in the virus. In the virus, we look for one of these genes. There are many assays looking at one of these genes, either the ORF1A, ORF1B, the spike gene, the E gene, the M gene, and the nucleocapsid gene actually of the virus. And actually now there are many, more than 65 commercial assays available for the detection of these genes. And all of these are approved by the FDA under emergency use. So they are not yet very well validated, but they are approved because the circumstances required this approval. So this link here, I don't know how you can get it, guys. Maybe we can put it on the Google on the YouTube, or you can just type FDA and COVID and you will get this list. So many companies has used either one, has made an assay targeting either one of these genes or more than one at the same time. So what are the samples that we take for the diagnosis of this virus? Of course, the best sample is the lower respiratory tract sample, so sputum or bronchoalveolar lavage. These are the best samples that are used, that can be used for the diagnosis or for the detection.
Sorry, Dr. Khalid, for interrupting you. There's only 5 minutes left for your explanation.
Yes, I need 2 minutes more actually.
Okay.
So we were talking about the lower respiratory tract samples as a better sample for the detection of the virus. If there is no sputum, then because many patients will have a dry cough, we can use the upper respiratory tract samples like nasopharyngeal swabs, oropharyngeal swabs, nasal swab, throat swab, and also saliva is a good alternative nowadays. So actually, a positive PCR, then when we get a positive result with this real-time reverse transcriptase PCR with the clinical manifestation of the disease, we can confirm the case. But when we get a negative PCR, unfortunately, we cannot preclude the presence of the virus when we have when the patient has symptoms. Why? Because the PCR sensitivity has only very high value at the beginning of the disease after the onset of the symptoms. So post-symptoms onset, it will have only 90% sensitivity. After that, by 5 days, the sensitivity of the PCR goes down to 80% after 5 days of the onset of symptoms. And after about 14 days of symptoms onset, it might reach about 50%. And the total PCR sensitivity is about 65% during the whole course. So you can imagine what are the consequences of this. Patients will probably not seek help early after symptom appearance. It will maybe go in this window, and then we are losing many patients. And this increases the probability of false negative results, and also this increases the load of infection control officers, and also increase the risk of transmission. So the other option we have for laboratory diagnosis is the immunoassays like ELISA. But as you know, antibodies develop later in the during the disease, not early in the disease, and we need to detect people early in the disease in the course of the disease. This paper showed that between day 1 and 7, and 38 of the infected patients will have total antibodies, 29% of them will have IgM, and these values will increase over time. So the IgM, IgG antibodies will appear sequentially in time. So, and they said actually, if we combine in the early phase of the disease, if we combine both the IgM detection and RT-PCR, we can reach about 90 or 95% of detecting positive patients. I think this is what I wanted to tell you about the virology part of the SARS coronavirus 2. I hope you enjoyed it, and hopefully we have a good discussion later. Thank you very much.
Thank you very much, Dr. Khalid. Thank you for your clear explanation about from the story, how this pandemic started, and classification, the name, also the transmission. A surprise is need to matter. It's also about the diagnostic. It is decrease the sensitivity of about PCR. Maybe we can talk next. And also I'm very interested because I'm structural biologist about the structure of virion structure on protein and entrance mechanism of this virus. So maybe next we will continue to understand SARS-CoV-2 at the molecular level. After that, maybe after this, we will combine together the Q&A for three keynote speakers here. So the next is continue to understand SARS-CoV at the molecular level. This lecture will be given by Dr. Wang Seok Mui. She's an associate professor and lecturer from the Faculty of Medicine, University Technology Mara, Malaysia. We call it UITM. She has amazing research interest in virus, including dengue and chikungunya, and focus on molecular diagnostic and antiviral screening. She got biotechnology bachelor degree from Royal Melbourne Institute of Science and Technology University in 1998, and PhD in medical biotechnology from University Malaysia Sarawak 2009, and got postdoctoral fellowship in medical microbiology University Malaya, and she has 31 peer-reviewed journal, also 3 books, chapter, and 4 copyright. It's an intellectual property of Malaysia. So next, the time is yours, Dr. Wang. Hello.
So,
maybe you
can you hear me? Can you hear me?
Yeah.
Okay.
Yes, we can hear you.
All right. Okay. Assalamualaikum and good day to everyone. Thank you to the Rector and Dean of Universitas Pembangunan Nasional Veteran Jakarta, and thank you to Dr. Yudi for the nice introduction. Okay, the topic that I'm going to present today is understanding molecular pathway of SARS-CoV-2. In this presentation, I'll be focusing on how the virus infects humans and leads to infection. This is the content of my presentation. First, I will give a brief introduction on the coronavirus, followed by its structure and genome organization. After that, I will talk on the infection cycle of SARS-CoV-2 and how it leads to COVID-19 cytokine storm and the potential therapeutic targets. Some of the information here have been presented by the first speaker, Dr. Khalid. However, I will briefly go through, so please bear with me. Okay, coronavirus are a large family of common viruses which are found in the human and animals. This diagram here shows a model of coronavirus evolution. Coronavirus in bats are the source of alpha and beta coronavirus, and coronavirus in birds are the source of gamma and delta coronavirus. To date, there are 7 human coronavirus which are present in the genera of alpha and beta coronavirus. The 4 circulating strains of human coronavirus shown in the blue box here, 229E and OC43, have been known for a long time since 1960s, together with the NL63 and HKU1, which were discovered more recently after the SARS epidemic. They are low pathogenicity, they're endemic and thought to be circulating in the human population. Approximately 10 to 15% of the common cold and influenza-like illnesses are caused by these viruses. The 3 coronaviruses in the red box here, the original SARS, MERS, and the SARS-CoV-2, which is the cause of COVID-19, are highly pathogenic and can cause more severe illnesses. These viruses emerge into the human population through species jumping or zoonotic transfer. SARS and MERS probably came from the bats, but it is thought that rather than directly jumping from the human to bats, they enter into the human population first through the intermediate host. For SARS, the intermediate host is thought to be civet cats and raccoon, and for the MERS, the intermediate host is most probably the camels. For SARS-CoV-2, it hypothesized that the virus would have jumped from bats to pangolin before jumping back to the humans in the local seafood market in Wuhan, China during 2019. However, there is no evidence to link the pangolin as an intermediate host of SARS-CoV-2 at this moment. Okay, SARS-CoV-2 stands for Severe Acute Respiratory Syndrome Coronavirus 2, and the disease caused by it is called Coronavirus Disease 2019, COVID-19. The virus may spread between people through close contact or via respiratory droplets produced by cough or sneeze, or indirect contacts via contaminated surface. Most people who fall sick will experience mild to moderate symptoms such as fever, dry cough, tiredness, and recover without special treatment. Severe cases may lead to shortness of breath, respiratory or multi-organ failure. The incubation period of the virus is around 2 to 11 days, with the median around 4 to 5 days, but may take up to 2 weeks to show the symptoms. The nasopharyngeal reaches the peak viral load approximately 4 days after infection and declines after that. The potential asymptomatic transmission has been suggested in several reports. However, the epidemiological studies have documented that pre-symptomatic viral shedding may have been the source of major infections. A person with subclinical infection shed the greatest amount of virus 2 to 3 days before the symptoms appear. Therefore, a substantial proportion of transmission probably would have already occurred before symptoms appear in the first index case. Risk of transmission is thought to be the greatest when patients are symptomatic, since viral shedding is the greatest at the time of symptom onset. However, the proportion of SARS-CoV-2 transmission in the population due to asymptomatic or pre-symptomatic infection compared to the symptomatic infection is not clear. Okay, now let's have a look at the structure at the structure and genome organization. Okay, SARS-CoV-2 is a positive sense single-stranded RNA virus. It is roughly spherical with a genome size around 30 kilobases. It has 4 structural proteins known as the spike protein, envelope protein, membrane protein, and the nucleocapsid protein. The nucleocapsid protein binds and holds the RNA genome. The spike protein, envelope protein, and M protein together, they create the viral envelope. SARS-CoV-2 lack of the heme agglutinin esterase, a glycoprotein that some coronavirus process to use for infecting the host. All right. So the genome of SARS-CoV-2 contains 14 open reading frame, okay, encoding for 28 proteins. The longest open reading frame is located at the 5' terminus, the open reading frame 1AB and open reading frame 1A, encoding for 16 non-structural proteins collectively involved in virus replication and possibility in the immune evasion. The 3' terminus encodes for the 4 structural proteins and 8 accessory proteins. Some of the proteins are involved in the viral pathogenesis. So, okay, now take a look at the structural protein in turns. S protein is responsible for allowing the virus to attach to the membrane of the host cells. The spike protein has 2 functional units, the S1 and the S2. S1 is the receptor binding domain. It binds to host cell receptor, whereas the S2 mediates fusion of the viral and cellular membrane. The receptor binding domain recognizes a specific receptor called the angiotensin converting enzyme 2 receptor, which is expressed in the lungs, heart, kidney, and intestine. Research has shown that spike protein of the SARS-CoV-2 bind to the ACE2 receptor with an affinity about 20 times greater than the SARS virus. This could be one of the explanations why SARS-CoV-2 is spreading so easily. Okay, M protein. M protein is the most abundant structural protein on the viral surface and defines the shape of the viral envelope. It involved in the viral assembly and interacts with nucleocapsid protein to encapsulate or to hold the RNA genome. Okay, moving on to the E protein. This is the smallest structural protein of the viral envelope. It is a transmembrane protein involved in the assembly and release of the virus from the host cell. During viral replication, the E protein is mostly localized at the endoplasmic reticulum and the Golgi apparatus. So essentially, the M and E protein play an important role in making the new viral particles. The E protein can also oligomerize and form ion channels that may play a role in releasing the virus. Underneath the surface is the viral envelope, which is derived from the host cell membrane. It is a lipid bilayer. Therefore, when in contact with detergent, the viral envelope will break down, killing the virus. This is the reason why hand washing soap is so important to prevent the spread of the virus. All right. Underneath the viral envelope is the capsid. Capsid is a shell that encloses the genetic material of the virus. Inside the capsid, we have the nucleocapsid or the M protein. This M protein is bound to the RNA, viral RNA, which contain the genetic information. The M protein appears to be multifunctional. It inhibits a lot of the host cell defense mechanisms and helping the viral RNA in replicating itself in creating the new viral particles. All right. Now I would like to talk about the infection cycle of SARS-CoV-2 in host cells. The infection cycle of SARS-CoV-2 in the host cell involves several stages. First is the host cell recognition and viral entry, genome replication and translation, viral assembly, and finally with the release of the virus. Okay. Angiotensin converting enzyme 2 or ACE2 receptor is a host cell receptor. It is a very well established receptor for SARS and SARS-CoV-2. SARS-CoV-2 gains entry into the host cells through the binding of the viral spike protein to the host cell, angiotensin to the host cell ACE2 receptor. Treatment with these anti-ACE2 antibodies can reduce the viral entry. All right. ACE2 receptors are expressed in lungs, artery, hearts, and kidney and intestine. It also acts as a peptidase that cleave the angiotensin 2, a vasoconstrictor to angiotensin 1 to 7, which is a group of vasodilator. Therefore help controlling the blood pressure by regulating the volume of blood in the body. The life cycle of SARS-CoV-2 in cells begins when the spike protein binds to the cellular receptor ACE2. The S protein is cleaved into the two subunits S1 and S2. S1 is the receptor binding. S1 is the receptor binding protein, whereas the S2 is activated by the host transmembrane protease serine 2 and results in membrane fusion. After the receptor binding, the conformational change in the S protein facilitate the viral envelope fusion with the membrane through the endosomal pathway. The virus is then released into the host cells. Once inside the host cell, the SARS-CoV-2 hijack the host machinery to transcribe, replicate, and translate its RNA genome and structural proteins. The newly synthesized structural and accessory proteins are then transported from the endoplasmic reticulum to the Golgi apparatus, after which the new virions assemble in budding the Golgi apparatus. Finally, the matured SARS-CoV-2 virions are exocytosed and released from the host cell into the surrounding environment to repeat the infection cycle. The released SARS-CoV-2 viral antigen subsequently bind to the antigen-presenting cells such as the macrophages, which in turn stimulate the production of a range of pro-inflammatory cytokines such as interleukin-1, interleukin-4, 6, interleukin-8, and macrophage colony stimulating factors, chemokines 10, tumor necrosis factor alpha. Ultimately, these cytokines proliferate and enhance. The massive release of the pro-inflammatory cytokines or cytokine storm leads to acute respiratory distress syndrome or ARDS. Okay, what is cytokines? Cytokines are a group of proteins, peptides, or glycoproteins secreted by the immune cells by the immune system. Okay, it plays a role in the regulating the immune response, inflammation, and formation of the red blood cell. Cytokines can be produced by a broad range of cells such as the macrophages, lymphocytes, leukocytes, mast cell, endothelial cell, fibroblasts, and a variety of stromal cells at the connective tissue. The type of cytokines produced depending on the functions and the cells that secrete it, which include chemokines, interferons, interleukins, lymphokines, and tumor necrosis factor. Cytokines can be divided into two categories: the pro-inflammatory cytokines, such as interleukin-1, interleukin-6, tumor necrosis factor alpha, which promotes inflammation, whereas the anti-inflammatory cytokines, which has the ability to counteract or inhibit the synthesis of pro-inflammatory cytokines. An excessive release of the pro-inflammatory cytokines will eventually lead to cytokine storm syndromes. Cytokine storm syndromes are a group of disorder representing a variety of inflammatory causes. The primary symptoms of cytokine storm are high fever, swelling and redness, extreme fatigue, and nausea. In severe cases, it may lead to multi-organ failure and death. When the immune system is fighting pathogens, cytokine gives signals to the immune cells such as the T cells and macrophages to travel to the site of infection. In addition, cytokines also activate these immune cells, stimulating them to produce more cytokines. In a healthy immune response, these defenders successfully contain and clear the virus without damaging the surrounding tissue and go on produce protective antibodies that can neutralize the virus. However, for reasons that are still not very fully understood, but may be caused by an exaggerated response when the immune system encounters a new and highly pathogenic invaders, the inflammatory process sometimes goes out of control. The overproduction of the pro-inflammatory cytokines called cytokine storm can cause serious damage to body tissue and organs. If a cytokine storm occurs in the lung, for example, in the case of COVID-19, the lung becomes so inflamed that it fills with fluid that patients struggle to provide enough oxygen to the body. This eventually leads to acute respiratory distress syndrome, ARDS, which is a life-threatening condition. All right. How SARS-CoV-2 leads to ARDS? How SARS-CoV-2 triggers cytokine storm that leads to ARDS? ARDS is a life-threatening condition. So in order to rescue the patient from this condition, it is important for us to understand how SARS-CoV-2 triggers cytokine storm that leads to ARDS. SARS-CoV-2 uses the ACE2 receptor and transmembrane serine protease 2 as cell receptor. Once the virus binds to the ACE2, both ACE2 receptor and the SARS-CoV-2 will be endocytosed. This reduces the amount of ACE2 receptor on the cells, okay, leading to an increase of angiotensin II in the blood. Accumulated angiotensin II induces inflammatory cytokines such as the tumor necrosis factor alpha and interleukin-6 through the metalloprotease 7, ADAM17. This is followed by activation of the interleukin-6 STAT3 target gene, which encode protein that drive the proliferation of cytokines. SARS-CoV-2 itself activates the nuclear factor NFKB via the pattern recognition receptor. So co-activation of NFKB and IL-6 STAT3 form a positive feedback named interleukin-6 amplifier, resulting in the excessive activation and production of cytokines which eventually leads to acute respiratory syndrome. So drugs or antagonists can potentially be used to treat the COVID-19. Drugs that block or interfere with the ACE2 receptor and the transmembrane protease serine 2 could help at the initial stages of the disease. A rheumatoid arthritis drug, which is called baricitinib, could inhibit the ACE2. Drugs like the chemostat mesylate, on the other hand, are known to inhibit the transmembrane protease serine 2. Anti-interleukin-6 receptor antibody called the tocilizumab and the tumor necrosis blocker, okay, could also suppress the function of the immune system. Chloroquine may suppress the cytokine storm, okay, by inhibiting the production and release of the tumor necrosis factor and interleukin-6, and also disrupt the membrane fusion during the virus entry. Okay. Alternatively, patient could also be infused with high levels of a soluble form of ACE2 to prevent the virus from binding the cells, a hypothesis that currently being tested in a clinical trial. Okay, so the treatment of patients with immunoglobulin from patients who has recovered from COVID-19 can also help with the immunomodulation. One interesting approach for treating the COVID-19 patient is to filter the patient blood through the special column that can trap the excess pro-inflammatory cytokines, thereby reducing their impact on the body. Hopefully, in the near future, a candidate vaccine will be available to treat the COVID-19. All right, actually, this is the end of my presentation. Okay.
Okay.
So yeah, so I would like to thank the Universitas Pembangunan Nasional Veteran Jakarta for inviting me, and University Teknologi Mara for the opportunity. Thank you. Back to you, Dr. Yudi. Okay.
Thank you, Dr. Wang, for your amazing explanation. Actually, I'm very grateful to get detailed understanding of this many things that I just understood just now about the structure and genome organization, infection cycle, and cytokine storm is very important for me, and also therapeutic target of SARS-CoV-2. There are some question here I would like to share, because the question is to Dr. Khalid and Dr. Wang. So directly I would like to ask you, from Dian Aruni from UN Sunan Kalijaga, Yogyakarta, there are some reported cases of SARS-CoV-2 infection from human to domestic cats. How could it happen? And there any possibility for reinfection or second infection from domestic cat that infected by SARS-CoV-2 to human again? So maybe after this, we can continue to pathological perspective and also community to Dr. Soroy. Could you answer this, Dr. Wang and Dr. Khalid?
Actually, to my knowledge, okay, maybe later Dr. Khalid can add on. Okay, to my knowledge, at the moment there are no concrete evidence that actually the cat is carrying the virus.
Yeah.
Right. So, Dr. Khalid?
Yes, thank you very much. Thanks, actually, Dr. Wang, for your very interesting talk. Yeah, there were few reports showing that the cat who was housed in the same house with a person who was infected with the virus also got the virus, but these were very few cases. There was a paper in New England Journal of Medicine when they did that experimentally, and they infected the cat with intranasally with the virus, and then they brought a cat also and housed that cat with the infected cat, and actually they found that the cat can get infected with the virus and infect a cat. But actually these were only 2 or 3 cats, I remember. They used 3 cats and they succeeded only 2 of them, they got the virus. So it seems it's possible. as Dr. Wang said, it is not very well evident. Many more studies need to be done on that. So they did that actually on the dogs. I think also it was not possible. So so far we know that the cat get infected, but still need to be confirmed. We are not sure whether now the infected cat can infect a human. That was not studied, and actually it was not reported. So yes, so we know that, as the question said, of course there are some reports, but it's not well confirmed. It still need a lot of studies to confirm it, probably, and experimentally, and also epidemiological studies. We have no report that the virus from an infected cat can infect a human so far. So maybe the virus that's produced is not suitable to infect the human. Based on the tissue factors, the different receptors, or something like that.
Okay, thank you, Dr. Wang, Dr. Khalid, for crystal clear for the answer the question. And also one more question is related to the molecular and virology approach. Is there any relationship between spike AC2 in an AC inhibitor? It's like a drug hypersensitive hypertensive. I'm not clear. It is the question from Lucia Sinchu Gunawan. Maybe someone.
Since actually the question is that since the ACE2 is the receptor for the virus, and the ACE2 is an enzyme, if we inhibit ACE2, can we inhibit virus infection? Unfortunately not, because as I said, the ACE2 is an enzyme, but the binding of the S protein to this enzyme does not need the enzymatic activity of this protein. So if you inhibit the ACE2, you don't inhibit virus infection. So if you inhibit ACE2 by any inhibitor against the enzyme ACE2, the virus still can bind to the ACE2 and still can get into the cell. So the binding and diffusion of SARS-CoV-2 to the ACE2 is irrelevant or does not need the enzymatic activity of the ACE2.
So my question next is, it is impossible to make the other inhibitor that can bind to AC2. Maybe the AC2 is not a great potential inhibitor, you mean?
Now there are two things related to ACE2, as enzyme activity and as a receptor, as a binding. So enzyme activity is irrelevant. Binding, of course, this is possible. People are doing that. And this is what Dr. Wang said about the antibodies. So antibody against the S protein will inhibit the binding between the S protein and the receptor. And also a blocking agent against that blocks the binding site of the ACE2 will also block the binding inhibit the binding between S and the ACE2. But unfortunately, it's not available yet. But if an inhibitor of the enzyme activity would have worked, it would have been marvelous. But unfortunately,
thank you, Dr. Khalid. Is there another comment from Dr. Wang? Dr. Wang?
No, just adding on to Dr. Khalid. Actually, at the moment, there are a lot of scientists. They are trying to find a way to block the spike protein on the virus by using the therapeutic monoclonal antibodies. So, but this is still on the way. So hopefully that in the near future, we can find a good monoclonal antibody can be used as to block the binding. Yes.
Thank you, Dr. Wang. So for the next is the last speaker. He will explain us about the clinical perspective and community of SARS-CoV-2. It is also the close important statement lecture from clinician. It will be given by Dr. Soroy Lardo, his internist, and also researcher at Faculty of Medicine, UPN Veteran Jakarta, and also he is a colonel, worked at Gatot Subroto Army Central Hospital. We call it RSPAD, Residential Hospital. So the time is yours, Dr. Soroy, since now.
Thank you, Dr. Yudi, as a moderator. Thanks, a good day. Hello, everyone. Thanks very much for coming along today. My name is Soroy Lardo from University UPN Veteran Jakarta. On this occasion, I will be talking about COVID-19, integrated clinical system, and tropical infection prospective community. Slide, Dr. The material topics consist of two. The first, clinical perspective, and the second, community perspective. Introduction, COVID-19, since it was declared a pandemic since March 12, 2020, has become a transboundary disease, not only as a viral infection with clinical manifestation, but has disrupted various sectors of life. The new strains of 2019 coronavirus were initially detected in 200 cases in Wuhan, China, by transmission to humans. Previously, we have identified the existence of MERS-CoV and SARS-CoV. The clinical course can be severe and cause organ disorders. The topic one, clinical perspective. We know that COVID-19 is a viral infection that can cause condition of acute respiratory infection with phylogenetic beta-cov Wuhan VDC, right picture and red box. As we know, the characteristic of a virus through its protein structure have the ability to susceptible host the replication, cytopathic effect, and cell penetration. Virulence and advancement of COVID-19 germs, the ability of COVID-19 is spreading infection, which can cause morbidity and mortality condition, namely ERDS. As you see here, COVID-19 has a protein, namely grabber, spuller, and cleavage after entering the lung receptor and replicates, disrupting the respiratory system and mutating. That threaten the patient susceptible to infection, comorbid, or immunocompromised patient. One of COVID-19 peculiarities has receptors in various organs. This shows that this virus has the ability to invade multi-organ with aggravating condition and even death. Our organization in infectious disease make a rule of three COVID-19 cause with increased mortality and community. Based on this, the influential role of three COVID-19 approach consists of clinical, including cytokine storm, ERDS, high affinity receptor, and community consisting late diagnostic, super spreaders, and quarantine failure. You see here, COVID-19 is clinical course, consists of three stages. Stage 1, early infection. This is a viral response phase. Stage 2, pulmonary phase. And stage 3, hyperinflammation phase. The stage is to facilitate the clinician in deciding COVID-19 management. Based on study in China, case stratification consisted of mild of 80%, severe of 13.8% were hospitalized, and 4.7% critical condition were treated in ICU. This slide shows the stability of the settled virus in the form aerosols, copper, cardboard, steel, and plastic. For example, the aerosol form has a half-life of 2.47 hours with a detection limit of up to 3 hours. How to enforce COVID-19 diagnosis? The discovery of clinical manifestation and contact history is very important. Prior to local transmission, the discovery of outbreak, site visit, and clinical symptoms is one cluster was supported by a CT scan with multiple patchy shadows and swab PCR confirmation positive. There is no exact choice to treatment, but antiviral vaccine plasma convalescence has been done. Supportive therapy according to the patient clinical condition and occurred complication. Chloroquine and hydroxychloroquine functions, the function to block the entry of viruses into cells by inhibiting glycosylation of the host receptor, proteolytic processing, and endosome acidification. This agent also has immunomodulatory effects through attenuation of cytokine production and inhibition of autophagy and lysosomal activity in host cells. Favipiravir is an active agent to stop viral replication by inhibiting RNA polymerase. COVID-19 understanding is determined by the process of incubation, like nasopharyngeal replication and asymptomatic, prodromal symptomatic but no evidence of pneumonia and hypoxia, and immunological with critical illness condition with multi-organ dysfunction syndrome and ERDS. An important note of diagnostic is the value of positivity in the swab. Nasal swabs can detect 2-3 cases and pharyngeal swabs detect 1-3 cases. A community perspective. Spread of infection that has spread between developing countries into community spread and local transmission. Some people are infected with no known source of infection, even without symptoms and transmission. The chain of transmission is determined by the droplet from coughing and touching hands, with cases surface contamination and then touches the infected person's hand and nose, mouth, and eyes so that it is infected with COVID. The picture shows the process of close contacts, airborne through inhalation or spray, and vomit to the surface, causing infection susceptibility. Based on the tracing symptoms of COVID-19 patients, consisting of anosmia, loss of smell, cough, fatigue, diarrhea, shortness of breath, and fever. The potential transmission from mild to moderate symptoms related to incubation, symptomatic, and recovery periods determined by detection of test coverage, isolation, and treating severe cases can prevent 60% spread. Based on the stratification of symptoms and the spread, it is very important to evaluate the symptoms. the spread of immune status is related to efforts to reduce mortality rates. This slide shows about the management of the isolation level is very important in collecting exposure data, identifying symptoms for clinical evaluation and treatment protocols. This slide shows the COVID disease pyramid. Pyramid 19 requires integration between health services and the community, namely infection control in addition to protecting the population with optimal care, prevention, intervention to the extent. In our country, the term for categorizing infected people consists of PDP, patient under surveillance, patient with breathing and fever symptoms similar to COVID-19 require hospital treatment, ODP, person under surveillance, person with mild breathing symptom without any symptom, and OTG, person without symptoms. Medical aspect OTG, ODP, and PDP. PDP has the characteristic of high virulence, high transmission, and the danger of reconnect. ODP has high virulence characteristic often not identified during the assessment. Rapid detection and isolation of the fever clinic are needed. Low virulence rate OTG with the potential to spread to vulnerable individuals, namely old age and low body immunity and infants. Our main step is how we comply with WHO recommendation with regularity and continuity of hand washing, wearing masks, health ethics, and social distancing. This slide shows the stages of hand and body hygiene continue. We must carry out like a 5-step for washing hands, before using and after taking off mask, before touching face after activity, and self-cleaning of bath with soap and changing clothes when at home. Medical aspect, epidemiology using mask and social distancing. The use of mask and social distancing is not 100% safe, but can protect us from the spread of droplets and aerosol. How about the self-isolation procedures for ODP? Requires disciplines for daily monitoring related to personal hygiene, nutritional adequacy, strengthening immunity and distancing. Thank you.
Thank you, Dr. Soroy, for nice explanation about the perspective, pathologic and community of SARS-CoV-2. So next, we'll continue to Q&A. For the first question, so I have to combine the question first from Ayodhya Haristiorini from UPN Veteran Jakarta. She asked about since SARS and MERS, it's actually for Dr. Khalid, SARS and MERS seems to be limited geographically. Why does SARS-CoV-2 have a higher transmission rate and more widespread globally? And the second one is also, is there any possibility that COVID-19 would continue to occur seasonally? I mean, only in summer or something. So maybe Dr. Soroy also can answer. And the first question, Dr. Wang also can answer.
So should I start or now, Dr. Wang? Should I start answering the first question? Okay. Yes, actually, that's a very interesting question. Why SARS and the MERS were limited geographically, and why SARS-CoV-2 spread globally? Actually, this can be answered at from different points. First of all, related to the virus. The SARS-CoV-2 virus seems to be able to transmit from human to human very efficiently for many reasons. First of all, because it seems that the virus has a very high affinity binding to the receptor, and this makes a small amount of the virus enough to initiate the infection. As Dr. Wang said, that the spike protein of the SARS-CoV-2 binds to ACE2 20 more with higher affinity 20 more times than the SARS-CoV-1. So there is better establishing of infection with a small amount of the virus maybe. The second point is that this SARS-CoV-2 replicates also to very high quantities in the upper respiratory tract. Okay.
Sorry.
Which makes it very easy to transmit from person to person, since the virus is present in high quantities in the upper respiratory tract. Also, actually, there is a large number of asymptomatic and pre-symptomatic transmission from the virus. And this also facilitates the transmission of the virus before even we can isolate, we can trace the cases and isolate them. So these are the factors that are related to the virus. There might be also some, for example, on the level of MERS and SARS, there might be some genetic predisposition that protected some people from the other viruses. It might be also the intermediate host, for example, the presence of dromedary camels here or there, that also localize the MERS to certain areas than other areas. Now, whether the virus is going to be, whether the SARS-CoV-2 is going to be a seasonal virus, there is a possibility, but actually, unfortunately, nobody can predict how the virus will behave. For example, SARS-1 just appeared and then disappeared after 2 years. SARS-CoV-2, we have to wait, but there is a high possibility actually.
Thank you, Dr. Khalid.
Okay, may I add on?
Yeah.
Okay, so the SARS and the SARS-CoV-2, they're actually using the same ACE2 receptor to gain entry. However, based on the research studies, why SARS-CoV-2 have more easily to transmit because there's a mutation in the SARS-CoV-2 virus that enable it to bind more easily to the ACE2 receptor compared to the the original SARS virus. Yeah, although they are using the same receptor to enter, but there's a single mutation in the SARS-CoV-2 that enable it to spread more easily, to gain entry into the host cells more easily. Yeah.
You mean this mutation is occur in
the virus? In the virus
itself, like in ACE2 or something?
Not the ACE2. It's in the virus itself. The spike protein. On the spike protein, yeah. Okay, just a single mutation, but it can change change everything. Yes.
Yeah. Okay. Thank you, Dr. Wang. Dr. Sore.
Okay. Based on my experience as clinician, 2 months manage patient with COVID-19, my opinions. Dr. Yudi, you hear?
Yeah, I hear just now.
My opinion, COVID-19, one of virus or a unique virus, and difference with dengue and HIV. But the first time, we have a lot of patients with ERDS, and after 2 months, we have most of patients with asymptomatic, that namely OTG, with positive result. Several patients positive until 60 days, but without symptoms. By my prediction, COVID-19 in the future maybe like seasonal flu or like HIV because of viral persistence, or maybe like typhoid fever as a carrier. We don't know in the next. This is my opinion as a clinician. And Dr. Khalid and Dr. Wang, please correct if I'm wrong for that. Okay.
Thank you, Dr. Saroy. So for the next question is, it's quite a deep question. The COVID-19 has a mutation in every country. As we know, we can see in nucleotides submitted in GS8, there's so many mutations. It's in each country, different mutation. Whether this is also implication for diagnostic and also the implication to the the symptomatic in every country is different, right? So what your comment about this mutation, various mutation in every country is differently. So maybe we start with Dr. Khalid about this kind of various mutation based on GSA.
Okay. So yeah, there have been a lot of mutations or many mutations reported in genomes from SARS-CoV-2, which were isolated from different countries. But actually, I'm not aware of any report that related these mutations to the severity of the disease. So if there is a higher mortality rate in a country to another, that not really depends on the mutation reported in the virus. Whether this has an effect on the diagnosis, yes, it might actually, but also there are no reports so far. But we have to be aware of that. So, yeah, we know that these viruses mutate, and usually when they design the diagnostic kits, they select in areas that are usually very conserved, so less likely to develop mutation in that area.
Yeah, I see. So yes. Next is Dr. Wang.
All right. From the diagnostic perspective, okay, because I also working in the lab for the COVID-19 testing. So one of the confirmatory gene is the RDRP gene that used for the confirmation of the infection. Okay, but based on our experience, we found that there is a mutation occur that affect the detecting of the test. And when we comparing by using a different test kit, I think because some of the primers commercial test kit that we don't know what is the primers sequences they are using, we realize that some of the maybe some of the samples like is tested negative by using one commercial kit but positive in another test kit. So I think that might actually, especially now, right, since the already a few months since the original the start of the infection until now, I believe there are some mutation that occur that will affect the in terms of the diagnosis of the virus. And another thing that I want to add on is the virus shedding. So by shedding of virus means there are two: shedding of the virus particle and shedding of the RNA.
Yeah.
Okay, so shedding of the virus particle is usually during the symptomatic phases. Okay, so shedding of virus particle is considered as contagious, then it can be transmit to other people. However, the RNA is non-infectious. So, but RNA, because it's not infectious, that is present in the body, it can be detected by PCR. So in Malaysia, so the Ministry of Health, we actually you have a new guideline whereby after admission of 14 days, 2 weeks, the patient can be discharged, although the PCR is tested positive. Because based on the research, they found that after a certain period of time, although they are tested PCR positive, but they are not infectious, meaning that they are shedding the RNA instead of the shedding of the virus particles.
Yeah, I see.
All right.
Okay, thank you, Dr. Wang.
Dr. Yudi,
Yeah, Dr. Soy, I would like to
Yes,
explain more.
I asked about how to implement about mutation with clinical field.
Yeah.
We have the patient, the first intubation with a mild condition. We isolate in our hospital, and the 6 days suddenly short of breath, ERDS, and the second patient suddenly acute renal failure that must be done CRRT. I think it's the condition of severity mutation or virulence of infected to the organs.
Doctor, sorry, I would like to ask you about the condition now in Indonesia, actually, because you work in RSPAD, right? So this mutation, is it correlate with the different symptom that only owned by Indonesian patient? I heard so many doctors say this, the symptom of COVID-19 in Indonesia is quite different with the other the abroad patient. So do you think is there any correlation between this mutation to the different symptom in patient of Indonesian patient?
In our hospital, we have cared about 350 patients. The first time, maybe 90% with ERDS, patient with comorbid, elderly, and heart failure. This is the first cases in our hospital, but now we have a daily practice, patient not severe, but with pregnancy with COVID, hemodialysis with COVID.
Okay.
Yes, and acute appendicitis with COVID. We prepare in our hospital 2 zones. The first zone, COVID zone, with OCAN pressure, negative pressure, 2 ICU, negative pressure for COVID, and ICU for non-COVID.
Thank you, Dr. Saroy, for a good comment about that. So next question is about still the mutation test question. Is it that since there's a mutation in SARS-CoV-2 that we know that already in every country, how about the vaccine? So it should construct based on the genetic, right, of the virus specific in every country, or how to make the potential or conserve which one we can make it, or do you any comment from about the case this question, Dr. Khalid and Dr. Wang, also Dr. Saroy?
I'm not familiar with the vaccine part.
Just a little comment is okay. Dr. Khalid?
Okay, I'll start.
Yes.
Yes. Actually, the mutation in the viral genome plays a role in the diagnostic and also in the development of vaccine. So actually, it will make the vaccine development a difficult task. But we hope that the antibody does not always bind only to certain epitope. It binds also in a conformational form to the protein. So we just hope that we get a good shot for the vaccine. Mutation that might affect the vaccine, yes, that might happen actually.
Okay. Thank you, Dr. Khalid. Next is Dr. Wang. Just a little comment about the vaccine or about mutation.
I think at the moment there are several candidate vaccines is available. They are still in the clinical trial. So in terms of the vaccine strategies, actually, I'm not very familiar. There are a few strategies actually. For example, by using the China one they are using the attenuated virus as a vaccine, and then some are using the particles, and then some using NKA, the modified Ankara. Can't remember. Okay, as the framework for the design of the virus, the vaccine. But I think this one I'm not very well versed, so I'm not So I
So
will give to
we
the next
believe
speaker.
that
Okay.
there is a term that make vaccine already think about this concern about the
Yes,
multi.
yes. There are several clinical trials at the moment, so we are still not sure how good is the vaccine. Yeah, hopefully it should be ready, but in the near future. Yeah.
Yeah, hopefully.
Yes.
How about Dr. Soroy?
Okay.
Yes, I hope the vaccine is available in the next. Indonesia collaboration, Biopharma and Eijkman Institutes now do to prepare about the vaccine. But my experience now, antiviral treatments with solidarity trial, WHO, and the second convalescent plasma in several hospitals in Jakarta, and now the region in Surabaya using convalescent plasma.
Okay, it is maybe the last question for us because it's already 3. Okay, first I will choose the question because we have limited time. It is from Desi Wenas ISTN. Are all baby born from the positive COVID-19 mothers can naturally antibody to face COVID-19 for the baby itself? Is it possible? I mean, she mean the baby born with mothers can naturally antibody to face COVID-19. Is there any case or information about that? Maybe Dr. Khalid first.
I'm not sure. Of course, if the mother was infected early before the pregnancy and then developed antibodies, IgG antibodies, these antibodies should be transmitted to the baby. As we know, the IgG antibodies will be transmitted to the baby. Now, whether this will to protect or not, most probably yes, but I'm actually not really, I don't have like a clear data about it to say yes or no, but theoretically, maybe yes, maybe Dr. Surai or Dr. Wang can help answering this question.
Maybe first, Dr. Wang.
I share the same opinion with Dr. Khalid. So actually, there are several cases already reported cases whereby the mother is infected with COVID-19, and the baby also have they have also the COVID-19 antibody. Whether this COVID-19 the antibody is protective against the infection, we don't have any data yet. Yeah, we don't have any concrete data. Yeah, okay, evidence. Yeah, okay.
Okay, Dr. Sarai.
Because maybe a little data about that that we don't know about if antibody as a protect baby from COVID-19. Yeah. The experience of dengue antibody is not protect if the secondary infection with another serologic type.
Okay. Okay, actually there's so many question now. I can see this in the comment or something. So, but the time is limited, so I have to close, unfortunately, this very, very good talk discussion. Maybe next we have to ask Dr. Khalid, Dr. Wang, Dr. Soro again to next discussion about this one, and in the other in the next future maybe. So, Excellencies, like ladies and gentlemen, maybe Bu Andri also asked me to close this this discussion. I'm very grateful again to the speaker for their excellent lecture, and also there's our agenda we have present to you this afternoon finish. It's been pleasure being with all you today. Thank you so much. Thank you so much for coming, for stay join with us today. We apologize if there are any mistakes we have made. Have a good day. Salam bela negara. Assalamualaikum warahmatullahi wabarakatuh.
Salam.
Waalaikumsalam.
Thank you very much. It was very, very interesting. A lot.
Syukran jazilan. Ana asatid li taqallum muruga Arabia, Dr. Khalid. Qalilan.
Assalamualaikum,
Dr. Khalid.
Waalaikumsalam, Dr. Surai. Thank you very much. It was very interesting.
Dr. Wang, thank you.
Thank you very much.
Dr. Wang, thank you.
Okay,
Dr. Andri,
Assalamualaikum. Bye.
Thank you.
Thank you, Dr.
Thank you,
Wang.
Dr. Yudi.
Is it possible
Thank you, Dr. Khaled.
We can make a picture together, maybe.
Yeah,
Thank you,
before that,
Dr. Wang.
nice.
Before we close, we can make a picture together.
Yeah.
Yes, of course.
Okay, I take
With salam negara, bela negara.
Salam bela negara.
Salam bela negara.
Maybe Dr. Khalid can like raise your hand and say salam bela negara.
Salam bela negara.
And Dr. Wang also.
Salam demi negara.
Salam bela negara.
Salam bela negara.
Bela negara.
Okay.
Salam bela negara.
Oh,
Thank you very much.
Thank you very much.
Thank you very much, Dr. Khalid, Dr. Wang, Dr. Sorai,
Thank you,
also
Dr.
Bu Andri.
Khaled. Thank you, Dr. Wang.
Dr.
Yeah,
Wang.
also Dr. Yanto.
Bye.
Thank you.
Thank you. Great.
See you, Dr. Soroy.
See you next time.
Yes.
See you next time.
Bye-bye.
Bye.
Goodbye.
Terima kasih Yudi, terima kasih
Thank you,
Bu
Dr.
Andri.
Yang.
Terima kasih Dr.
Terima kasih, Dr.
Soroy.
Yeah,
Ya, terima kasih semuanya.
For anybody who wants to.
Terima kasih teman-teman semua ya.
Sorry, Dr. Khalid.
Is the YouTube recording available for anybody who wants to see it later?
Yeah, of course. We already recorded. I will send you the link.
Okay, great. Thank you very much. Then I can send.
Okay, thank you. Bye-bye.
Bye.
See you then.
Assalamualaikum.
Assalam.
Salam.